negative. There are 5 megabases of DNA in E. coli's genome, 4.2 megabases of DNA in B. subtilis, and 1.8 megabases of DNA in Streptococcus thermophilus. And plasmids are naturally occurring in all 3 species. So E. coli is, of course, found in the human digestive tract. Bacillus subtilis is found in the camel digestive tract, horse digestive tract, and also in soil. And Streptococcus thermophilus is found in yogurt, of course. And an interesting story with Bacillus subtilis and the discovery of Bacillus subtilis, while during World War II, some German scientists were in North Africa, they and their colleagues started contracting dysentery and getting very ill and occasionally dying because of it. And so, but they realized that the native tribespeople of the North African area were building up an immunity to the disease. And so they followed them around for a day or so and found that these tribespeople had an interesting method of doing this. They would find a camel and follow it around, and then when the camel laid feces, they would pick up the feces and consume them. And the German scientists were able to isolate from the camel feces what we now know as our lovely Bacillus subtilis. So, some of the advantages of using Bacillus subtilis: it takes pure genomic DNA for integration without modification. It does not require any linear DNA. Its chassis has been shown to work with very large amounts of DNA, approximately 4 megabases. It can be sporulated for long periods of time and is very resilient. And its natural competence means simple transformation. It's also very good at expressing proteins, and it's generally regarded as safe. So, yeah. Disadvantages: it's not E. coli, so you can't use MAGE, no simple point mutations, or bacterial artificial chromosomes. It's not Saccharomyces cerevisiae, so it's not completely orthogonal from other bacterial cells, and it's not as well characterized as either of these organisms. And integrations require at least 125 base pairs homology, rather than only 30 to 40 base pairs. And Bacillus subtilis is less efficient at DNA transformation. So now I'm going to hand over the mic to Keone Gandel to discuss some more of the design of the experiment.
Okay, so our experimental design, we really wanted to make a system where you didn't have to modify the DNA you wanted to put into it to use it. And so what we used is a positive and negative selection cassette. What you did simply is we had bacillus homology on our plasmid that would integrate into the bacillus cell. Then on the 2 sides of the plasmid would have the LP sets. Those are the landing pad sites. Those are user defined and will vary from organism to organism, or from whatever you want to integrate. Once you have that integrated, you can take a raw DNA strand, such as the standard backbone with an RFP, and since it has the homology, you can directly add that to the bacillus competent cells. It will integrate it, and since it has the negative selection cassette in here, you don't have to actually select with an antibiotic. You can just have whatever part you want. And that is really the biggest advantage of our system. Since you define what you want to put in, and you don't have to modify the DNA that you're originally putting in, it really has unlimited application. And in the literature, it's been shown to easily integrate over 300 kilobase pairs of bats and other genomic DNA, including a mitochondrial genome of the mouse. So, and since it's high throughput, and since it's standardized, you can potentially put the entire biobrick registry into sporulated cells for long-term storage. So our experimental design. What we did is we created about 5 different verified plasmids. We have other constructs that we're currently working on. These were called the 4 YINT plasmids, which stand for yeast integration, and then PG base, which is going to be a general plasmid to use. These are designed so it would integrate over the terminus of the cell. I'll get to why that's important later. But they have sites for integration of raw genomic DNA from yeast cells. And I'll go over the positive negative selection cassette here. The reason it's innovative is because the negative selection cassette includes a toxin under the IPGG promoter. This gram-positive one is very well regulated, and so will express almost none of the toxin when you don't have the induction conditions. And then it also has spectrumized resistance. And so your entire construct will integrate over this. YINT 5 kb will integrate a 5 kilobase pair fragment from the yeast genomic DNA. YINT 20 will do a 20 kilobase pair. YINT 100 will do 100 kilobase pair, and YINT 1000 will do 1000 kilobase pairs, or 1 decabase pair. So first step in our project would, of course, be integration. What you would do is have your Bacillus subtilis cell and with your little landing pad sites, and then have a genome of what you would want to put in. This could also be a DNA plasmid, it could be a BAC, it could be raw genomic DNA from a mammalian cell, it could be anything you define with the landing pad sites. Then you go integration, and it will integrate the entire construct over the terminus. The reason that's important is cells are actually structured. So up here at the top, there's an origin of replication, where the replication fork goes. But down at the bottom, near 180 degrees from that is the terminus. The terminus actually terminates replication of the strands. And an interesting study was shown where if you integrate a large amount of DNA on one side of the cell, it will actually make the cell grow much slower or kill the cell. But if you put more DNA on the other side to even it out and have the terminus 80 to 800 degrees from the origin, it will survive. This is because, of course, it stops and the replication becomes lopsided. But by removing the terminus from the bacillus cell, we defeat those size limitations. The next step would be, of course, excision from the chromosome. You have your 2 combined cells, but what you really want from them is yours. You're just using this, the bacillus as a chassis to help you carry your DNA or modify it or construct it. So we were thinking of using Krelok's-mediated excision. So these would separate the 2 genomes into your S. thermophilus, or your guest genome, and then a Bacillus subtilis cell. And then next would be selection. We would destroy the hybridized genomes, any of them that are left, and also Bacillus subtilis, leaving yours for possibly a new cell, a minimal cell, or downstream purification. So some innovations we had. First of all, we used the positive and negative selection cassette for integration rather than deletion. In other literature, they use it for deleting certain genes. We also replaced the terminus to defeat size-limited integration, unlike the paper where they integrated a cyanobacteria genome. We are also using bacillus rather than yeast for full genome integration, faster growth, easier storage, better stability, and easier for bacterial labs to use. Progress. We have verified that 20 kilobase pairs of raw genomic yeast DNA can be integrated. No modification required. We cloned modular plasmids for this assembly, such as the YINT ones and the PG base, and we also verified current methods for Bacillus subtilis integration. Future prospects, I'll go over this quickly. We want to integrate S. thermophilus successfully and successfully show excision, integrate other bacterial and yeast DNAs for proof of concept, and integrate possibly the entire BRICS registry into cells. And some other things, we want to do serial integration and rigorous optimization. We have a lot of roadblocks. S. thermophilus would actually not grow from ATCC. They arrived dead. But we were actually able to verify that S. thermophilus was in yogurt. And by using colony PCR, we could verify that the S. thermophilus strain we actually wanted was in the yogurt. But we still wanted this fully sequenced strain to work with. And then YINT 100 and even 1000 kb didn't successfully integrate. But we verified successful integration of the into 20 kb. Here are some of the cells. You can see E. coli, bacillus cells, and then these guys are the Aspergillus that we found. STK6 bacillus cells weren't very competent, so we used a protocol from another lab where Munich. The gels were also very messy. We weren't sure if it was a buffer problem, but we were planning on rerunning the gels. And each time we wanted to transform something, we'd have to make new cells because we didn't have access to a negative 80-degree freezer. And finally, there's also difficulty with the PCR machine. Since we're a hacker space and we don't have as many resources as many other hacker spaces or as many other labs, our PCR machine didn't heat up enough to actually DNA the DNA for our PCR reaction. We used a thermometer to figure this out, and we also began using another PCR machine because all of our PCR machines are very old, 10 years. And the main thing is we couldn't actually give our project to the parts registry without putting it in our own vector because the positive and negative selection said the negative selector is unstable in E. coli and will kill the cell. You have to have another gene, it was somewhere else on that plasmid, inside of the standard backbone for it able to replicate in E. coli. And then I'll hand it over to Dan for acknowledgements.
These are the people who helped us, and we should thank them. We wouldn't be here without them. And let's talk about human practices. We're rich in human practices because we're in a lab in downtown Los Angeles, very close to East LA and about 2 blocks away from Skid Row. So there's literally 10,000 homeless people within 2 blocks of us. It's a challenge. A member's car was vandalized the other day, and it's a challenging environment in many ways. Our next door neighbors in our building, actually we shared a wall with them, a piece of drywall, and we had a lot of water coming underneath that piece of drywall, and we found out they were having a marijuana grow with hundreds of plants. We thought, well, some grows are legal, we give them the benefit of the doubt. The police didn't see it that way, and they raided it when Corey was there one day, and luckily they got the right door. Our door was right next to theirs. So we've had our challenges, but we accept everyone in our lab. That's our human practice. We have not too many members, but we accept all, and these young people have found us. We're not exactly sure how they found us, but we welcome them. We have some top-notch scientists like Corey and others who come to our lab, but we accept everyone, so that's our human practice. We collaborated with Art Center in Pasadena. They hosted us over there, and we let them use our lab for part of their experiment about 2 weeks ago. And then Santa Clara Law School did the human practice, or the policies and procedures part of our wiki, and they're here as well. So that's how we collaborated. And we work in a very small community of DIY biolabs. I see people I've known from before, from BioCurious and other labs. We keep in close contact on mailing lists, and we met each other before. So it's a very small world, and we do collaborate with each other. And we look forward to collaborating with anyone else here who's interested in our project. Thank you.